I'd like to start by thanking MIN, the NIJ, and Pittcon for inviting me to come here. I'm really excited to talk about what we've been doing in the area of forensic epigenetics. The project began, I suppose, with a court case I was working on. And this was in Michigan in the early 90s. A woman was found murdered. There was trace DNA under her fingernails. It matched her husband. There was a custody dispute between her and her ex-husband. And at the time, the last time she was seen alive, she was handing over their child to the husband. The DNA, the husband could not be excluded from the intimate mixture of DNA, and there was a presumptive test for blood, but it wasn't confirmatory. The suspect argued that it wasn't blood. The actual DNA was from secondary skin cells transferring from him to the child to the woman, and that was why DNA from the husband was found under her fingernails. Critical to the determination, was it rust or blood? Was it human or animal? And even more importantly, since I was to testify on this case 20 years after the incident occurred, could the sample be tested many years later? And it got me to thinking about all of these different issues in terms of body fluid identification, and that started it. It also, we had a wonderful explanation of the different issues with current serological testing based on a variety of presumptive and confirmatory tests, some of which are very useful and other ones are very complex and time important. So the basic idea is that there are a variety of ways if we want to do next generation body fluid identification. Very nice talk on the aspects of the proteome. Other researchers have been looking at transcriptomes, RNA, using reverse transcriptase PCR or capillary electrophoresis. But we've been looking at DNA methylation because we feel like the methylation is at the root of the genome tree, and we don't have to worry about differences in expression and other aspects that might have to come into play in other methods. Because we're using DNA procedures, we can use PCR and we can be very sensitive. So what are the advantages? The best thing about the epigenetic project that we have is it's very compatible with existing DNA processing. The sensitivity is high. We're nanogram to sub-nanograms levels of sensitivity. Our primers are human-specific. The extracted DNA that we're using can be used for multiple purposes. It's very simple to go back and take a sample of DNA that's been extracted and put in a refrigerator or freezer and go back and test it for body fluid specificity. There's one disadvantage that we have to deal with, and that is the methylation sites must be identified prior to amplification because methylation isn't preserved by PCR, and I'll talk more about that. Generally speaking, it should be obvious to every one of you that your human body, even though all of your DNA is exactly the same, it exists in many different forms of many types of cells, skin, hair, teeth, etc. But your DNA is all the same. And how does your body differentiate cells? And even more importantly, I've known for a long time that twins had different fingerprints. And also, those of you that know twins know that over time, their appearance begins to change. So what is that? And that's really the basis and the roots of epigenetics. The idea is that there are heritable differences in our DNA, and they're related to a variety of different factors that are present. The one we're interested in, of course, are differences in methylation, which affect gene transcription, and there are also effects like histone modifications and other things that can be used. And in general, epigenetics is the study of these heritable changes. Most striking example, of course, would be the transformation from the caterpillar to the chrysalis to the butterfly. I've always been fascinated by that as well. So it's all about gene expression, and in terms of methylation, the methyl residues are covalently bound to the 5' carbon of the cytosine, and that's really neat because that means that we have a covalent bond. It's going to be very stable. The turning on and off is based on methyltransferases, and one of the other very interesting things about DNA methylation is that it generally occurs upstream of genes. It controls gene expression, and we have this discussion of methylation of the cytosine. in terms of what are called CpG islands or CpG dinucleotides, and 70% of CpGs are methylated invertebrates. And you see these distinct patterns which control gene expression. So these CpG islands are some of the areas that we're looking at. We want to find regions of gene silencing or gene activation based on methylation. As you can see, in this particular case, muscle cells versus epidermic cells are quite different, and it just really depends on whether or not there's the methylation pattern, as you can see there, which would turn on and off different aspects of the gene and the protein. So how do we exploit this forensically? Let's find locations in your genes that target the expression of cellular proteins or things that are specific to different kinds of cell formation. How will we find these things? Well, we can look at patterns. We can look at studies that other people have done with RNA or with protein, or we can do whole genome array studies, and we can piggyback on the back of cancer studies and other all kinds of different things. So we don't have to start from first principles. And we can look in the genome for tissue-specific methylated sites. So that's what we usually call them. And look at differences in methylation that are dependent on cell type. So the very first thing that you can do, and you can see in the literature, or the scientific literature, you find a lot of people have done whole genome array studies for cancer or for other types of genomic studies. And if we can find particular locations that are very specific to different cell types, we can exploit that. And there are arrays presently produced by Illumina that have 850,000 different sites across the whole genome. And we can use these, if we can target them and do a little bioinformatics, maybe we can find some information. But to start with, we looked at this one study by McCord, who did a cross-correlation of various kinds of cells and their global methylation differences. And as you can see, there's differences are displayed by color. And sperm, of course, is very different from skin cells, the keratinocytes, or lymphocytes, etc. And we can try to find, and it's kind of a treasure hunt. It's really fun, because you look through these sites and you try to find locations which are specific to one particular type of cell and not others. Now, how we do it, there's a variety of different ways. You can find enzymes that cut specifically at different methylated sites. You can find antibodies. But the best way we found to do this is looking at bisulfite modified PCR, because what it does is, if the cytosine is not methylated, the addition of the bisulfite creates a change. First it sulfonates it, and then modifies the amine up there, and you get a uracil. If the methylation is present, there's steric hindrance, and you don't get any addition. And so you can lock in place, once you do this reaction, these two different forms of the same seed. But what the interesting that happens next is shown in this slide. The Cs are converted, if they're not methylated, to uracils. But if they're not, and you can see that in the red, that 1C, if it's methylated, it's not going to convert. And so it gets carried on, and then we have what really, essentially following PCR, looks like SNPs, single nucleotide polymorphisms, at various locations. Anywhere a C existed that wasn't methylated, it will change. Okay? So the next step is to, knowing these things, we look at specific genes, or we look at specific locations, and we design primers in and around those places upstream of the genes, and we assess the candidate CpGs. So the big problem, of course, is finding the ones that are specific to one particular cell type, and it involves a certain amount of screening. It also involves primer design. You have to design the primers around the fact that some of the Cs are going to change to Ts. And if you do it correctly and you get a nice clean amplicon, then you can move further. So this is a microfluidic chip that's doing an analysis. And finally, we use pyrosequencing. And it's interesting because originally we hadn't the capability to do pyrosequencing. We had thought that the method that we would use was real-time PCR, because we'd had some experience with high-resolution MEL, and we knew that the AT base pair, of course, has two hydrogen bonds, versus the GC one has 3, and we thought there'd be MEL differences. But we found, to our dismay, that that wasn't quite as easy as that. And luckily, we hooked up with some people at the University of Southern Mississippi who had a pyrosequencer, and later got one ourselves. And the advantage of pyrosequencing is that it produces a very precise signal and allows you to determine the absolute quantity of difference of methylation. I think I can show this if it will come up. Okay. I don't know if you can hear it. The basic idea is it's a cascade reaction of enzymes. If you add a base, you produce the pyrophosphate. The sulfurase converts it to ATP. The ATP then is interacting with the luciferase and converts to light. So you get a signal because you added a G. The next base that you're adding, there's a series of 3 of them in a row. And so the signal for those 3, since we're adding 3 nucleotides, is 3 times as high. And then we get a signal the same way. And in effect, at one particular location, we're either going to have a C or a T, and we can determine the relative percentage by what gets added, and that's how it works. So our objective then will be to find some markers that will differentiate biofluids, design primers around them, and then perform sequencing reactions with the pyrosequencer. We had done some work with respect to McCord. I had a fabulous student, Tanya Mahdi, who went through the literature and found some locations, and she was very lucky or good, I think both, and in finding a small set of markers that were very discriminatory. So the methodology, as I mentioned, was extract the DNA the way you do in a normal way. You do the bisulfite modification for a subset of those samples, and then you can either do PCR or real-time PCR. Here's Tanya right there. The DNA is extracted and modified. Some of the loci she selected, based on, one of them was FGF7, identified as a fibroblast growth factor, hypomethylated in skin cells, and also hypermethylated in semen, which was kind of unusual because we often find semen to be hypomethylated. The next one was a BCRA4 gene. Actually, we were expecting this to be hypermethylated in sperm, but it wasn't, and it didn't work. But strangely enough, and interestingly enough, it was hypermethylated in saliva, and maybe related to the fact that you can use saliva for a genetic test for BRCA. Other markers that we've looked at, FGF7 for skin cells and semen, and finally, you get to see some of the effect of this. So here's a marker for blood and semen, and blood is hypomethylated. Blood is hypermethylated and 95% C. At the same location, the semen is 2% C. So a very distinct difference between the 2 different types of body fluids. So we had a whole set of markers that Tanya had developed. There's C20, BCAS4, Z3, and FGF7. And each of them are specific. There we have one for blood, one for saliva, one for semen. And the FGF7 is interesting in that it has intermediate levels also for blood, semen, and saliva. So useful for maybe a multiple test. We also began working, of course, with Dr. Bala at the University of Southern Mississippi, and he did some additional bioinformatic work and found additional markers for blood and for semen. The next step, once we performed all this work, was to start to sort of get types of validation. And there's some very specific ways that you do validation in forensics. You need to look at things like sensitivity, age, specificity, degradation, and mixtures. And we did all of those things. One of the first things we looked at was sensitivity, and we found sensitivity varied from about 100 picograms to somewhere around 10 nanograms, depending on the primer design and some other things. Later on, through collaboration with the San Francisco Police Department, we actually discovered that it had to do with the fact that we were amplifying a little too fast. And it was interesting, they were able to drop our sensitivity an order of magnitude or more simply by slowing down our amplification, because we had other projects we were interested in fast amplifications. Another thing that we saw was something called PCR bias when we dropped the samples to really low. And this is partly due to the fact that, again, this effect of how fast we're amplifying, and also due to effects based on specific CPGs and primer design, creating differences. You can see that with the black bands, a little bit more error in certain CPGs than others. So we had to be careful for that, and we carefully assessed the CPGs. They're numbered 1 through 7. They're 1 in a row, and we've, of course, collected the ones that are most specific. One of the big questions we had was, what about really old samples? So here's a 20-year-old sample. They're essentially indistinguishable. Both blood and semen were looked at. And also, with the assistance of the San Francisco Police Department, we found that our marker is actually specific for sperm, not semen, which is useful too. With the assistance of Dr. George Duncan, we looked at a whole bunch of different samples to see if if we were human specific. We found a few of them amplified with our primers, but there's a set of 3 primers. There's primers for the PCR amplification and then a sequencing primer. And when you add all 3, we weren't able to get any response except from humans. With one slight exception, we had to deal with large primates, just like he does. And what we found is that for chimps and gorillas, we could still get results, but not orangutans. And I'm not sure what the significance of that is. Another question that we had often answered is, could you tell the difference if you had a mixture of blood and semen in your sample? And the answer is absolutely. There would be an intermediate level of methylation. Degraded samples could still be amplified as well, and there wasn't a marked effect based on that, other than whether or not they could be amplified. Okay, so the next step, once we finished that first step of analysis, was the thing that we were lacking and the thing that we wanted the most were vaginal markers. It's very important in sexual assault. We also wanted to go back and look at the real-time PCR assays and a couple of other issues in terms of phenotype and age. Jokingly, Dr. Duncan always tells me, well, your holy grail is the detection of the vaginal epithelial, because it's a crucial point of evidence for sexual assault and child abuse. But the problem is that epithelial cells found in the mouth vaginal skin are very closely related. There has been some research in terms of looking at the microflora of the vaginal, but we wanted to see if, so we did a really thorough study and looked at some more array studies, in particular one by a group in South Korea, and we found one location, PFN3. As you can see, this particular marker is intermediate in methylation between blood, saliva, and semen. And it, with a P less than 0.05, we can use it as a marker for the vaginal epithelia. Again, it works well in mixtures. We can look at mixtures of vaginal epithelia and semen and get intermediate levels if it is present. The other thing that we started to think about with this was, could we go back to real-time PCR? Because it would be a faster, simpler way to do it. Maybe not as specific, but something that could be easily done even if a lab didn't have a pyrosequencer. And of course, as I mentioned, the difference is that when you have an AT base pair versus a GC, which is what we're basically generating when we do the bisulfite modification, you can see differences in melt temperature. And so if the DNA strand is methylated, there's no bisulfite conversion, and you'd have a higher TM. If it's unmethylated, it would take a lot less energy to pull it apart. The big secret, of course, is finding locations where there's a huge difference in methylation between, say, a semen and the other types of body fluids that might be present at your crime scene. So we can do that in this particular case with semen because there are certain markers we have where it's very low levels of methylation. So we published this paper on a quick identification of semen based on real-time PCR as well with melt curves. And Joanna did a lot of this work and was also funded by a grant from the National Institute of Justice, a scholarship. Once we started doing that, Joanna had this idea, well, maybe we just haven't found enough markers with specific differences. So we began to work with the bioinformatist at FIU, Dr. Yuri Narasimhan, and he developed some programs to look at the array data from different kinds of array. This is the one from Park, and we're doing some ourselves now at FIU, and where we can categorize the virtual differences by examining a whole variety of different types of body fluids. Once we do that, we begin to find new markers, and we have a suite of new vaginal epithelial markers and other blood markers that we're beginning to develop. So we're real excited by that work as well. And finally, the last part of it that I really like to talk about was kind of an aside, but it turned out to be just as important, and that's the determination of age. And I talked to you about gene expression, and so and people often ask, well, are epigenetic changes related to environment? Some of them are. That's very true. But of course, obviously, if your epigenetic changes are related to gene expression, they're going to be very robust. So we've been looking at the robust ones. Now let's look at some that are less robust. And in fact, we want these changes that aren't even affected by environment. What we're looking for are epigenetic locations, which maybe over a period of time stochastically become methylated. So it's a gradual, slow process of increase in methylation. And these are the things that are going to affect how you look and how your body behaves. So we began to look at that, and we thought about the importance of age because many of you are aware of the, again, efforts in terms of identification of people. And I found a website on the site that could actually artificially age you. So I artificially aged my daughter, Melanie, and it kind of worked. You know, she's looking a lot older there, but it also gave her a beard, so I don't think it worked that well. But in actual fact, Deborah, who was collaborating with us from Brazil, was able to locate some new places. And there's been a lot of research in this area because there's a question of biological versus chronological age. So we went through and looked at some specific sites. And I also have another new student who is also working on this. And we found a number of locations which are age-specific and can be identified based on, and you can determine plus or minus, or an average difference of about 7 years. And some people have done better than that and others worse. But we're very excited by that. But one of the most interesting things about this work was that it demonstrated that the age specificity varied with the type of tissue. So the other thing that we needed to do was, and we're starting a new grant, is looking at combining body fluid identification with age. And we're really excited because we expect that we can find different markers for saliva, blood, semen, and vaginal epithelial in terms of determining what a person's age is. And so more specific analysis that way. The other thing that we've been thinking about, and it had to do with my thinking about cell phones and forensic, the computer forensics or digital forensics. And I'm sure you're all aware that if someone take apart your cell phone, they could probably figure out where you've been over the past few weeks, or maybe over the whole life of the phone. And we began thinking, well, what sorts of things could you determine that aren't genetic but are related to your lifestyle, and maybe they would deal with epigenetics. Many of you may be familiar with the Dutch famine, and the babies that were born in the year of, I think it was 1944 to 45, when Germany cut off all food to the Dutch. There were issues that continued even after the babies were born, and babies that were born from those babies had differences in birth weight depending on whether their mother was starving the whole time or part of the time. And so we were figuring that things might be there, and sure enough, in the literature you can find things like smoking, alcohol intake, body mass index, and maybe other things, exposure pollutants that could be related to your lifestyle. So again, maybe we can start to find markers to read your DNA like a book. The first one that we've located just recently is an epigenetic biomarker for smoking, and it's shown here. This is by looking again at chip array studies, and we found one particular marker in the AAR gene that had a loose correlation with smoking, and we drilled down, Hussein did, looking at a whole variety of different sequences around this particular gene, and came up with some very specific markers in blood for the determination of a smoker, and again, a little less specific in saliva, but still pretty good. Okay, so in conclusion, we're using epigenetic methods. They're at the root of the gene expression tree. We think they're very valuable for that purpose. They can be used to discriminate blood, semen, and vaginal epithelia. We have other loci we're looking at as well, sweat, etc. Methylation shows great stability in samples stored for up to 20 years. Mixtures of body fluids produce intermediate results, and phenotyping methods have been discriminated for age and tobacco smoking, and there's results in the literature also for, of course, body mass index and other things. The course, the acknowledgements, the major support for this work was provided by the National Institute of Justice. We also had help from the Pontifical University, the Catholic University of Rio Grande do Sul in Brazil. We have just recently a postdoc from the Seoul National University, their Department of Forensic Science. We're collaborating with the San Francisco Police Department, the Broward Sheriff's Office, and of course the University of Southern Mississippi. I also want to thank QIAGEN for helping with support with our pyrosequencing work, and thank you all for listening to me.